when we think about evolution in microbial communities, we have a lot to learn from that of experimental evolution studies, such as those conducted in the lab. So, uh, Lenski and colleagues showed that evolution can be rapid and repeatable in test tubes, in populations of E. coli grown in test tubes. Now, these are simple communities consisting of one species. And the question is, how does evolution proceed in complex communities, such as the human gut microbiome? Here you have hundreds of species, not just one, all interacting, competing, and collaborating. And it's unclear whether ecological processes might supersede the evolutionary processes, especially on short time scales. And so one might ask, what are the mechanisms by which the microbiome responds to selective pressures? Uh, One way could be purely through ecological responses like species fluctuations over time. So here we see these, species abundances colored in these colored balls shifting over time. And surely we have seen these kinds of responses in healthy versus diseased individuals. And so this is something that has been well documented. But if we were to go ahead and sequence individual members of our individual species of the community, you'll see that there's a tremendous amount of genetic diversity within these species. So you'll see that there, for example, are multiple strains of the same species coexisting. And this can give rise to a substantial amount of the genetic heterogeneity. So here, what I'm showing you is time series data collected over 1 year. This is data from Poirier and Grusin. And we, in our lab, phased strains belonging to the species Bacteroides vulgaris. And you can see that there's strain fluctuations over time. And these strain fluctuations could actually be another ecological response to selective pressures. Alternatively, you might get strain invasions. So maybe a resident strain lacks all the relevant genetic material to survive in a changing environment. And so an outside invader bearing some adaptive variant might come in and colonize the host. So that's yet another ecological response. But finally, we might see evolution. And here, evolution is where you have a genetic modification of a resident strain, either via mutation or recombination. And I just want to really underscore that this is an innovation. This is something that doesn't already exist in the system. So this is quite distinct from a strain fluctuation. And till recently, it was unknown whether evolution actually occurs in the microbiome, especially of healthy human hosts, which has been largely the subject of my study. And the reason is, in the absence of any selective pressure, why should they evolve? And moreover, a lot of these species have been coexisting with us for millions of years. And so maybe they are already optimally adapted to us. Now, studying evolution in the microbiome is a challenge because the problem is we want to track mutations arising on a specific genetic background and not confuse this with strain fluctuations. And that's something that I'll get into a little bit more deeply in just a moment. So why should we look for evolution in the microbiome? Well, microbiome genetic variants play an important role in our health. They're implicated in the digestion of food. They can confer antibiotic resistance, drug metabolism, affect the way we metabolize drugs, and even confer gut inflammation. And one could potentially take, so the issue is that I'm showing you pretty much the handful of examples that we have in the literature as to how microbiome genetic variants impact host phenotypes. Yet there are obviously billions of mutations and genomic sites to inquire and find out what their functional potential are. And what my former mentor, Katie Pollard, likes to say, we're kind of in the era of the year 2000 when the human genome just came out. We had all this DNA sequence, but we didn't know what each gene or SNP was doing. And so we're kind of in this dark age right now, with a lot of promise of discovery ahead of us. So I argue that if we take an evolutionary lens to understanding what these variants are doing, we may be successful. And concretely, if we find sites that are adaptive, we might identify what sites are actually really relevant and important for our health. So thus far, we've been talking about different ecological and evolutionary processes that can give rise to potential responses to selective pressures. We've talked about species fluctuations, strain invasions, and evolution. And today in my talk, what we're going to look at is evolution on two timescales. We're going to look at evolution within hosts operating on matters of days and months, and then we're going to look at evolution across hosts, looking at timescales potentially exceeding our own lifetimes. All right, so let's jump in and take a look at what's going on within hosts. So my lab largely looks at evolution using whole genome shotgun sequencing data. And the reason for this type of data is it's very ubiquitous, it's very affordable, it's very high throughput. It allows us to sample, hundreds of species simultaneously. But it has its drawbacks. It's basically a mishmash of all species, and the data is represented in these short 100 base pair fragments. And so we need to somehow figure out a way to grapple with this really messy data such that we go from this to identifying mutations that are at high frequency. So one way we deal with the heterogeneity of the data, given that it comes from so many species, is we map reads to reference genomes. And so you can map the red reads to the red species genomes, and etc. And so then you get polymorphism frequencies, as like I'm showing you here. You have this frequency at 50%. Now, people have been employing these strategies for now almost a decade, and you can, in this way, look at genetic differences between samples. So for example, if we look at this blue species across many different hosts, you could look at one site and see, oh, this site is at 80%, 30%, 0, etc. And people have been quite interested in using this kind of information to perform strain tracking. So for example, using this genetic information, you could identify strains that are shared between mothers and their infants on the basis of genetic similarity that is unique to that dyad and absent from the broader population. And then, of course, we in our group have been looking at how to estimate proportions of where our microbiomes come from. For example, Leah Briscoe in my group has estimated that a significant fraction of our microbiomes when we are in the NICU as babies come from floor and isolate top, etc. Okay, but going back to this question of detecting evolution from shotgun data. So what we are looking for concretely are mutations that go from low to high frequency over time. And we're looking in the data set that we're going to be looking at, we're looking at relatively short time scales of 6 months. And so these large frequency changes are inconsistent with drift, basically, and more consistent with adaptation. And also looking at these large frequency changes are helpful in avoiding detecting sampling error, allele frequency changes due to sampling error. Now, there's a problem. We might actually have multiple strains of the same species co-colonizing the same host. And remember this diagram I showed you, you have multiple strains coexisting, and a frequency change of these strains could result in genetic changes also accruing over time. And those changes are not due to evolution, it's just due to this flip-flopping of these strains. So without going deeply into how we address this, we, in brief, basically resolve the population structure to be able to distinguish evolutionary events from strain fluctuations. So in other words, we look for a dominant strain present in our samples that we can confidently genotype and then track its genotypic changes over time. It's kind of like in PopGen when you're studying humans or fruit flies, you want to control for population structure before you start to do any kind of analysis looking for adaptation. So, so the spirit is the same, just the techniques are a little bit different. Okay, so having done this, we then turn our attention to some data, which is 250 healthy hosts from the Human Microbiome Project. And these hosts are sampled over 6-month timescales. They've not had any kind of obvious selective pressure like antibiotics or shifts in diet. And we're going to track the evolutionary changes in this set of hosts and see what we find. And so on the x-axis, what I'm going to show you are the number of SNV changes. And on the y-axis is the fraction of comparisons with n or more SNV changes. And so this is the data we get. So if we look at the roughly 30 most prevalent species across all the hosts that we have around 800 data points. And you can see that this line starts at around like 10 or 11%. So it turns out that the vast majority of our pairs show 0 SNV changes on the order of 88%. Okay, so nothing seems to be happening, but then you see that on this side of the plot, about 5 or 10 SNV changes accrue in about 9% of our populations. And then on this side of the plot, you can see that around 10,000 SNV changes accrue in about 3% of our populations. So you can see that there's just vastly different orders of magnitude of SNV changes accruing, either 5 or 10 versus 10,000. And this bimodal distribution suggests that there may be 2 processes resulting in these 2 different amounts of SNV changes. And those 2 processes are strain invasions or fluctuations and evolution. So let's unpack this a little bit more. So let's say we take a strain of E. coli from each of us, and we drew a tree describing the evolutionary time that has elapsed between those strains. If you had a strain invasion from, let's say, your neighbor into you, then you'd expect a tremendous number of SNV changes to accrue because of the amount of time that has passed between those 2 strains. By contrast, if we take a look at the SNV changes accruing within a host over time, then just because there's such shorter amount of time elapsing, you would expect far fewer SNVs to accrue. So going back to this plot, we can contrast what's going on within hosts with that of between hosts. And you can see that between hosts on average have 10,000 SNV changes, consistent with this long amount of evolutionary time that has elapsed. So, so what we have then is evolution out here on the left side, consistent with few number of SNV changes, and invasions on the right side, consistent with what we see accruing between hosts. So what we are showing is that yes, bacteria do evolve in the host gut, and evolution, is not this rare occurrence that occurs on long time scales. In fact, it can occur on short time scales right alongside those species fluctuations. Ecology and evolution can happen simultaneously in the gut. And once again, this was relatively unknown finding as of 5 years ago, mainly because this was, it wasn't really fully known at the time if commensal bacteria in the absence of a selective pressure would evolve on such short timescales. And so what this implies is that this is kind of a dynamic that we cannot ignore, even while we continue to probe species fluctuations, we must study evolutionary changes alongside. So we and my colleagues, Benjamin Good, Oscar Halachek, and Catherine Pollard, wrote a paper about this work in 2019, and alongside with us, Shiji Zhao, Tammy Lieberman published their paper looking at the same kind of phenomena using isolates. And soon after, many papers have come out showing the pervasiveness of evolution in human commensal gut bacteria, using a variety of techniques like high-C sequencing, long-read sequencing, etc. So now that we're establishing that, yes, evolution does happen in the microbiome, we can start to ask other more nuanced questions, like how do within host microbiome ecological and evolutionary dynamics change over our lifetimes? In the work I just showed you, we were looking at healthy humans that were adults. And so now we can contrast what was going on in adults with that of perhaps infants. And so alongside my former student, Daisy Chen, we asked, do the evolutionary dynamics differ between that of infants and adults? We speculated that it would, mainly because infants have different diets, different immune systems, and even different microbiome community members. So in this work, we analyzed more than 700 publicly available infant shotgun metagenomes coming from these papers. These infants, first the mom was sampled at the time of birth, and then infants were sampled over the first days, weeks, months of life, and then the first year. And we were able to contrast what was going on in these infants with that of healthy adults from the Human Microbiome Project. And so what we're going to show is on the x-axis, different epochs in life. So this is the mother to infant transition. We treat mom as one time point and infant within the first week of life as another time point. We're going to ask, what evolutionary changes accrue as the strains transmit from mom to infant? Then we're going to look at what's happening in the infant during the first week of life, the first month of life, the first year of life, and then contrast that with adults sampled twice in 6-month timescales. On the y-axis, we're going to look at the SNV changes per day, per base pair. And so this is what we get. We get this plot where you see this rapid decline in the rate of evolutionary changes. And in fact, as the bacteria transmit from mom to infant, it evolves more than 100 times faster than what you see in adults, stable, healthy adults sampled on 6-month timescales. So the rate of evolution can in fact be incredibly rapid, and perhaps this is a mechanism for bacteria to survive in their novel environment. We asked further, what is the modality of evolution in these infants? Are sweeps seeded by de novo mutations or recombination events? De novo mutations are spontaneous mutations that rise to high frequency. Recombination seeded events are as follows. Well, I'll show you in a second, but we saw limited evidence for de novo mutations, mainly because we saw actually a tremendous amount of purifying selection across hosts. It doesn't mean that they're not happening, but instead we actually think recombination seeded sweeps might be more common. And this is what they might look like. You have a donor strain carrying your favorite gene 1 and a recipient strain carrying your favorite gene 2. An HGT event could then result in gene gain and loss. So we can go ahead and take a look in the infant data to see if we get any gene gains and losses, and indeed we do. In fact, we see tremendous activity of gene gain and loss, and moreover, this seems to be very rapid initially and then decline over the first year of life, similar to the dynamics seen at the SNV level. So HGT is emerging as a very important evolutionary force acting within us. And we are not the only ones to notice this. And of course, we're not the first ones to notice how common and important HGT is. Several other important papers are coming out, such as, for example, Benjamin Good's paper with Zira Liu, looking at the dynamics of recombination within hosts. So many of the sweeps within us seem to be seeded by recombination events, and we're going to return to this point a little bit later in the talk as we start to consider how sweeps might be transmitted across hosts. Finally, I wish to share what happens to strains themselves. Some of them actually might be replaced early in life. A strain gets in, and then it might get replaced quickly soon after. And so you see, once again, this incredible amount of dynamism early in life giving way to stability in adulthood. And so in a recent paper, what we showed is that, in fact, adult microbiomes are really stable. And we quantified this using a stochastic logistic model, which unfortunately I won't go into at the moment due to time constraints. But essentially, you can see that, yes, over the course of a year, the strains that you have are the strains that you will have tomorrow. But these are one-year timescales, and you might consider, well, what happens over really long timescales? We're looking at the arc of life, where early in life you see a lot of dynamism giving way to stability, but there are limits to the stability. We might want to ask, how long can we hang on to those strains that we have today? The ideal data to test these long-term dynamics is longitudinal samples over a few decades. But obviously, this is very challenging to come by. So what we're going to do is instead use data from adult twins to test for these long-term dynamics. Work by Corpella et al. Showed that cohabiting teenage twins and siblings share many strains. And there happened to be this data set by Shi et al. That had sampled 125 adult twin pairs that grew up in the same household and then lived for 40 years apart before being sampled. And so we asked the question, do adult twins share the same strains? And the answer is no. So what that implies is that you may have the strains that you have today, and they may be actually quite stable, even in the face of antibiotics, remarkably. But eventually, over a decade or two, you will lose all the strains that you have today. Why that is and when that occurs is an open question. Okay, so to summarize this part of the talk, dynamism gives way to stability, but only for so long. So now we're going to consider a longer time scale than what we have been considering already. Instead of looking at evolution within hosts, either on daily time scales or over our lifetimes, we're going to look at evolution across hosts. And this may encompass time scales that exceed our own lifetimes. So thus far, we've been looking at sweeps occurring within hosts, and we might want to ask, do the same evolutionary changes arise in multiple hosts? This work is done with Richard Wolff, a PhD student in my lab, very, very talented. And so when we ask this question of, do the same evolutionary changes arise in multiple hosts, we looked in the literature, and we actually didn't see much evidence of parallelism occurring. Remember, in fact, we see a lot of instances of purifying selection. So de novo mutations don't seem to be arising again and again across many hosts. Instead, maybe one person evolves a genetic adaptation, and then that strain migrates to many other different hosts. However, this is inconsistent with the data because if you look at different hosts, they actually have different strains, very, very different, distinct strains. Remember, we looked at number of SNF changes between hosts, and we saw they were on the order of 10,000 SNF changes out of a million total base pairs. That's about a 10 to the minus 2 divergence, which is the same order of magnitude divergence as humans and chimps. So this picture of the same strain colonizing many, many hosts is not really consistent with the data. So instead, what we think is happening is that there's migration and recombination. So this kind of mechanism would not only pass around the adaptive fragment, but also preserve the genetic diversity along the backbone. Remember, we saw a tremendous amount of horizontal gene transfer within hosts, and so it's plausible that HGT is playing a role in transferring material across hosts. How would this work? Well, you have a host with a strain and its adaptation, and the strain might transmit to another host, and there might be HGT, and that HGT event will then result in attractive homogeneity across hosts. Okay, so we've noticed in the literature again and again that in fact antibiotic resistance genes can be passed around using this kind of mechanism. So the question we have is, how commonly are gut commensal bacteria genes passed around in the same manner? So how do we detect selective sweeps across gut microbiomes? So here what I'm showing you are haplotypes from different hosts. This is host 1, host 2, host 3, and scattered on here is a beneficial variant. Then you have neutral variants in gray, and then you have a deleterious variant in red. As the beneficial variant rises to high frequency, it will drag with it the linked variation to high frequency as well. And you all may be quite familiar with this already. It's a signature of a selective sweep corresponding to a region of elevated linkage disequilibrium. And of course, this kind of signature has been exploited in population genetics for decades, especially when applied to organisms like humans and fruit flies. But to date, this signature has not been really leveraged in bacteria for reasons that we're still trying to understand. But I think one reason is that the pervasiveness of recombination is still becoming appreciated. And so, thus far, approaches to look for selection in bacteria have largely focused on looking at signatures of, let's say, DNDS and so on. So here what we're going to do is we're going to look for these selective sweeps by looking for regions of elevated linkage disequilibrium. But as you know, other non-selective forces such as demographic fluctuations or low recombination regions can elevate LD too. So what we're going to do to try to address these confounders is we're going to propose a new selection statistic called ILDS, or Integrated Linkage Disequilibrium Score. What this statistic does is it compares LD between non-synonymous sites, which are presumably under selection, versus synonymous sites, which are presumably neutral. So this is how the statistic works. At the onset of selection, the beneficial variant may be rare. As it rises to, and also deleterious variants will also be rare, and they'll be rare because purifying selection is tamping down their frequencies. There's so much purifying selection acting in the genome, you'll likely have a rare deleterious variant private to the haplotype that the beneficial variant is on. So as the beneficial variant rises to in high frequency, the linked deleterious variant should also rise to high frequency in very tight LD. Of course, synonymous variants will also rise to high frequency too with that beneficial variant, but they may be in less tight LD just because they've had more time to segregate onto different haplotype backgrounds. And so if you frequency match the synonymous sites versus non-synonymous sites, you should on average see higher LD between non-synonymous sites compared to synonymous sites in a selective sweep. We can test out our intuitions using simulations, using the software SLIM. And so we simulated neutrality, and here you have LD between non-synonymous sites shown in red versus LD between synonymous sites shown in blue. And you can see under neutrality, we don't see a difference in that LD, but under positive selection, we do. All right, so now we're going to test out our statistic using a pathogen called C. difficile. And this causes a lot of gastrointestinal disorder. And there's a locus that's under purifying selection called TCDB or toxin B. And so we're going to ask can we detect this positive control using ILDS? Here is a scan where on the x-axis is the genomic position, the y-axis is the ILDS value, and this is what our scan looks like. You can see there are lots of peaks, and in fact, they correspond to regions of significance, concretely where you have these LD between non-synonymous sites being elevated compared to LD between synonymous sites. Okay, it turns out that one of our tallest peaks is in fact TCDB. And so this confirms that in fact, our statistic is quite capable of detecting positive controls. It turns out that these other peaks, several of them, coincide with other toxin factors, some of which have been hypothesized to be under positive selection. So not only are we able to pick out what is known to be under positive selection, but we can confirm what hasn't been shown to be positively selected as yet. All right, so now we're going to apply the statistic to Ruminococcus brommi, a gut commensal bacteria. And this is data from the Human Microbiome Project. And you can see that we pick out a locus called MDXCF. MDXCF is involved in maltodextrin metabolism. What is maltodextrin? Maltodextrin is this additive added to highly processed foods like chips, sriracha, baby formula, etc. And so we hypothesized that, in fact, our diets are playing an important role in driving selection in our gut bacteria. To test whether this maltodextrin metabolism might be due to our westernized diet, we then took a look at data from the Unified Human Gastrointestinal Genome Database, which consists of publicly available metagenomes converted into MAGs from more than 30 studies from around the world. And so we took a look at westernized communities, they're colored in gray, these are their sample sizes, versus non-westernized communities in orange, to see if the sweep at the MDXCF locus is specific to westernized communities. And in fact, it is. So here is a sample of some of the scans from the westernized communities, and you can see that this peak shows up again and again in westernized communities, but is absent from the non-westernized communities. So this suggested that, in fact, our westernized lifestyles are playing a crucial role in shaping the evolution of our gut bacteria. If we take a look at other species, you can see that there are pervasive sweeps across many species, and in fact, we see an enrichment for carbohydrate metabolism genes, once again in line with the hypothesis that diet is playing an important role in their evolution. And here are some scans in other gut commensals. Maltodextrin metabolism pops up again. We see starch utilization systems genes showing up. These are shown to be evolving within hosts as well. And then we also see transcriptional regulators. So what I hope I've been able to convince you is that evolution is pervasive, not only within hosts, but also across hosts. And across hosts, it seems to be mediated by migration and recombination. Okay, in the last couple minutes, I'm going to touch on another important evolutionary force shaping gut commensals on long time scales, surpassing our own lifetimes. And this work is done with Jonathan Ma, PhD student in my group, and Kirk Lohmuller, my colleague here at UCLA. And so we recently wrote a paper on the inference of the demographic histories and selective effects of human gut commensals over the course of human history. And this is a little cartoon showing what we're about to tell you. So I'm going to talk about a little bit what is a distribution of fitness effects. Essentially, this is a quantity that estimates what will the fitness effect of a new mutation landing in a genome be. So this is an example of an SFS, of a DFE that we infer. On the x-axis, you have the selection coefficient going from neutral to deleterious, and the y-axis is the proportion of sites bearing that selection coefficient. In this example, you can see that the vast majority of mutations are in fact quite deleterious. Okay, so how do we infer these kinds of quantities? So these are haplotypes, once again, similar to what I just showed you, where you can have mutations at different frequencies across different hosts. You can bin these mutations into a quantity called the site frequency spectrum. And so you get singletons in this bin, doubletons in this bin, tripletons, etc. Then you can infer demographic history, which can shape the height of these bars in the SFS. And then you can also infer a distribution of fitness effects, which can also influence the heights of these bars. And so one important assumption about SFSs is that they actually assume independence between sites. Remember that we actually see a tremendous amount of recombination happening inside the gut microbiome. And so we are going to actually use this SFS with the assumption that, in fact, recombination is sufficient to unlink sites. Okay, so how does the SFS help us infer a demographic model or DFE? So once again, here's our SFS. On the x-axis is the prevalence of an allele. The y-axis is the proportion of SNPs falling in each bin. And for a constant population size, each bin should have a size of 1 over x, where x is the prevalence. Now, if you have an expansion, a population expansion, such as shown here, the population size increases from past to present, we expect an excessive rare variance. Negative selection should also result in an excessive rare variance, because mutations are being tamped down in frequency. However, a contraction in population size should result in an excessive common variance, and then balancing selection should also result in an excessive common variance. So you can see that selection and demographic changes can have confounding effects. So what we're going to do is we're going to actually focus on synonymous sites in this work because and try to infer a demographic model from that with the assumption that the synonymous sites are neutral. And then once we do that, we're then going to try to fit a DFE to the non-synonymous sites, controlling for what underlying demographic model may be influencing the shape of the SFS for both non-synonymous and synonymous sites. Okay, so we're going to now, after having built our SFS using the synonymous sites, we're going to infer the demographic histories, and this is what we get. So case example, this is Ruminococcus bromi, this is its SFS, and if we were to infer maximum likelihood estimate of its SFS, you can see there's a really great fit. And when we fit a model, and this is all using the software FitDaddy, you can see that we infer actually a population size expansion, as shown on the x-axis. This is the ratio of the current population size versus ancestral population size. And we infer it to have happened roughly 57,000 years ago. Okay, so we can do the same thing for all the species in our data set. On the y-axis, we have the time in terms of years as to when the demographic change occurred. On the x-axis, we show whether there was a contraction in the population size on the left side of this red line versus an expansion in population size. And this is what we get. So this is the species we just looked at, Ruminococcus bromi. But you can see actually that on average, these species kind of span this arc of history of humans. Many events are occurring 1,000, 10,000, 100,000 years ago. And remember that 10,000 years ago was the agricultural revolution. 100,000 years ago was the Out of Africa movement. And we also have more recently things like the Industrial Revolution. And so while of course it is tempting to speculate that some of these events may be coinciding with human events, like for example, agricultural revolution, for example, this species involved in the degradation of starches, future work will actually connect these demographic histories with human activity. Um, there is one paper I wish to highlight by Cornejo et al., which in fact was successful in connecting a demographic history for the cavity-causing bacteria Streptococcus mutans, which was inferred to have a really large expansion coinciding with the agricultural revolution. So many of these histories may in fact be tied with human activity, but once again, that's something that is for future research. Okay, so I just want to emphasize that these models are a starting point. And in fact, there's a lot more work to be done to refine them and to infer more sophisticated models. Okay, so now this brings me to the finally inferring the DFE of these gut commensal bacteria. And so we're going to once again look at Ruminococcus bromi, and remember we inferred this model, and now we're going to throw in the non-synonymous variants, and you can see that there's an excess of them compared to synonymous sites, indicative of purifying selection. And so we can infer a DFE based on this non-synonymous SFS, conditional on what we inferred with the demographic model, and we get this shape that I already showed you, where we have a large amount of mutations that are expected to be highly deleterious. We can infer this across many species, and in fact, you might see that there are some trends that look to be driven by phylogeny. And so these species, for example, seem to have very similar looking DFEs compared to, and these might have similar, less deleterious DFEs. And when we, in fact, quantify how similar the DFEs are within genera versus between genera, we see that DFEs within genera are in fact more similar, suggesting that these DFEs are more related than not. And then finally, we looked at core versus accessory genes, and we see that they might have more deleterious DFEs than accessory genes for some species, which makes sense given that core genes are more fundamental and present in every single lineage as opposed to accessory genes. And so we expect that there might be potentially stronger selection coefficients among core genes, but of course we also infer lower effective population sizes, and so another force acting here could be drift. So thus far, I've been showing you DFEs inferred looking across hosts, and I think a next step for the field is to actually look at DFEs within hosts. And it could be that there are fitness trade-offs across host environments that, for example, could explain reversions. You might have an A in this host and a T in this host, and that could just be a reflection of the varying fitness landscapes across many hosts. Okay, so this takes me to my conclusions. We've looked today at evolution on two timescales. We looked at evolution within hosts, we looked at evolution across hosts, and within hosts, we see that bacteria can evolve within a matter of days and months. We see that dynamism gives way to stability, but only for so long. We see across hosts that pervasive selective sweeps across is mediated by migration and horizontal gene transfer. And then we see varied demographic histories and DFEs across species. And with that, I'd like to thank the very many people in my lab, as well as former mentors and colleagues and collaborators and funding.